Hello, aspirants. In the previous module, we generally see what is analysis and what is analytical chemistry and its applications. In this module, we are going to specifically see about pharmaceutical analysis. Though more or less both modules talking about the same information, the pharmaceutical analysis is more specific here, and the kind of analytical techniques and analysis we perform in the pharmaceutical industry is specifically spoken about in this module. Let us go through this module to understand pharmaceutical analysis. What is pharmaceutical analysis? Pharmaceutical analysis is traditionally defined as analytical chemistry dealing with drugs both as bulk drug substances, that is active pharmaceutical ingredients, for example paracetamol active substance, and then the finished pharmaceutical product, that is formulation, for example paracetamol tablet. And also pharmaceutical analysis or the analytical chemistry which deals with the packaging materials which is used for the pharmaceutical products. Now, why a pharmaceutical analysis is required and what are the stages the pharmaceutical analysis is required, which are important to understand in research and development as well as the manufacturing industry in pharmaceuticals. So during the formulation process, either in the research and development or manufacturing units, the raw material will be purchased from the approved qualified vendors. So these raw materials should be tested, analyzed for their quality and appropriateness. So before purchasing these materials, raw materials, the vendor qualification will be done by the quality assurance personnel of the specific R&D or the manufacturing pharmaceutical company to evaluate the quality of the raw materials. Once the raw materials is qualified by the quality assurance department of the purchaser or the R&D or the manufacturing company, the supplier will supply the raw material to the R&D or the manufacturing unit. Then the raw material once procured, it will be tested for its potency and physiochemical property because it may vary from lot to lot. So every time you purchase it has to be tested. It may vary. Hence each lot of the samples need to be analyzed before use. And once the raw material received from the vendor, sampling needs to be taken to perform raw material analysis. The material under analysis shall be kept under the quarantine area until the analytical report is released. As we saw in the previous module, the pharmaceutical analysis also classified into qualitative and quantitative analysis. Qualitative pharmaceutical analysis is the determination of those elements and compounds that are present in the sample of unknown material. So you name the elements and compounds from an unknown material. Just name, that is qualitative analysis. Or you determine a structure, or you are going to see that the molecular weight, something like that, which is qualitative in nature. Then the quantitative analysis is the determination of the amount by weight of each element or compound present in. For example, assay, which is a quantitative analysis where you are determining whether the paracetamol tablet is having 500 mg of the label claim present in the tablet. That is a quantitative analysis. So to understand the pharmaceutical analysis and instrumentation in a better way, we need to understand some of the important terminologies which is used in the pharmaceuticals. First one is active pharmaceutical ingredient, which is defined as the substances are intended to furnish pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment or prevention of a disease. So basically you are treating a fever with a paracetamol tablet. So the paracetamol tablet will have the active pharmaceutical ingredient called paracetamol. So the tablet will have the paracetamol is the active pharmaceutical ingredient and which is used to reduce the fever. So the active form of the particular tablet which is named as the paracetamol is called as active pharmaceutical ingredient and the pharmacological action is reduction of the fever. So these active pharmaceutical ingredients can be manufactured or can be synthesized by different process including chemical synthesis, fermentation process, recombinant DNA or other biotechnology methods or isolation recovery from natural sources or any combination of these processes. Chemical synthesis, for example, the paracetamol or ibuprofen are being synthesized by a chemical process. Fermentation, for example, many antibiotics like cephalosporins or penicillin are manufactured or synthesized through fermentation process. And some of the biologicals or protein or peptides are being manufactured by recombinant DNA vaccines, also used to manufacture by the biotechnology methods. Then isolation recovery from natural resources, for example, digoxin is obtained from the natural sources. And this entire process can be used in combination to produce a particular active pharmaceutical ingredient. What is the definition of a drug? So drug is an article intended for use in the diagnosis, cure, mitigation, treatment or prevention of disease in humans and other animals. And articles intended to affect the structure of or any function of the body of humans or other animals. So which is the definition is similar to active pharmaceutical ingredient which we saw in the previous slide. Now what is drug product? Which is a finished dosage form, for example a tablet, capsule or a solution that contains an active pharmaceutical ingredient generally but not necessarily in association with inactive ingredients. For example, the paracetamol tablet will have an active pharmaceutical ingredient paracetamol and also it contains lactose, magnesium stearate, polyvinyl pyrrolidine, etc. can be inactive ingredients which are used to bring that paracetamol into a tablet form. So that particular finished product can or may not be having these inactive ingredients. The term also includes a finished dosage form that does not contain an API but is intended to be used as a placebo. So those product or a finished product which do not have the API which is called placebo. Now what is intermediate? A material produced during steps in the synthesis of an API that must undergo further molecular change or processing before it becomes an API. The same way applicable for drug formulation also. So before we get the finished API, active pharmaceutical ingredient, we can get a intermediate. That means from the source material, the active pharmaceutical ingredient is synthesized. In between, it is transformed into a different chemical compound. So from the source material, a chemical compound is transformed and from that chemical compound, API is synthesized. So in the in between, there is a chemical compound called intermediate. Those intermediates also being supplied by many manufacturers to the synthesis of the final active pharmaceutical ingredient. So the middle compound, the compound which is used for the synthesis of active pharmaceutical ingredient and it can be the precursor for the synthesis of active pharmaceutical ingredient is called intermediates, but it need not to be the starting material for the particular synthesis. Then it becomes to impurity. What is impurity? A substance that is present in small amounts in another substance, making it dirty or of poor quality, or it can be toxic also. So these impurities are can be produced during a manufacturing process, or it can be a byproduct of the starting material also. And These impurities can be formed during the storage period by the formulation excipient interaction, or it can be leached or extracted from the packaging materials as well. In pharmaceutical industries, analysis is performed in different stages of the process, or the development process, or the manufacturing process. So in the R&D and the industries, the analysis involved in many stages, such as during a manufacturing of the API, active pharmaceutical ingredients, or synthesis of the API. And once the API and excipients are received for the formulation development, those APIs and excipients also analyzed for the appropriateness or particular selection for the formulation development. Then during the formulation development, the formulation will be analyzed intermediately for their quality as well as the performance. So if the quality and performance in between the development is not good, the formulation again redeveloped. And once the formulation is finalized, the final formulation also will be analyzed for its quality and performance. Then the packing materials which is used for the packing of the particular drug formulation also will be analyzed. So basically, during the procurement of the API, production of the API, then the procurement of excipients, then during the development of the formulation, then the finished product formulation after the manufacturing, then the packaging material selection as well as the packaging material finalization. These analyses are performed in the development and manufacturing of the pharmaceutical products. In a pharmaceutical analytical laboratory, the story of the analysis starts with the R&D analytical laboratory. If it is a API or a pharmaceutical finished product, the analytical method will be developed for the raw materials and finished products. The analytical methods will be developed for assay, dissolution, related substance, impurity analysis, etc. for examples. Then these analytical methods which are developed will be validated for the finished products like active pharmaceutical ingredient or the tablets or the capsules, whatever the formulation which is developed for different parameters as I explained before, like assay, dissolution, etc., whichever is appropriate for the specific dosage form. And this process involves preparation of standard testing procedure for the particular methods, then the validation reports, stability reports, etc. Then finally, these methods are transferred to the quality control laboratory, which is in the manufacturing unit. So in the R&D stage, we are developing the product. During the development, we are developing analytical methods for the raw materials and finished product and validating it. Then we are transferring the method to the manufacturing. So these methods will be used in the quality control laboratory to ensure the manufactured product is having the appropriate quality and performance. So once these methods are reaching this quality control laboratory, method verification will be carried out for the specific method. Then the raw materials and the packing materials used for the manufacturing will be analyzed. Then the finished product finally will be analyzed by using this developed method which are verified at the QC. Then finally the finished product will be analyzed and the report will be prepared for the batch release or the quality release of the particular product for the marketing and distribution. What are the analyses carried out for different kind of pharmaceutical products or pharmaceutical excipients or the active pharmaceutical ingredients? So the API will be analyzed for identification, assay, water content, related substances, impurities, microbiological testing if required, particle size analysis wherever appropriate, and if required, other testings as appropriate. Then the excipients will be analyzed for identification, assay, water content, related substances, impurities, microbiology if required, viscosity, pH, conductivity, whatever is appropriate for the particular excipient. Then the finished product, whether it is a tablet, capsules, ointment, gel, ophthalmics, injectables, oral liquid, suspension, whatever it will be analyzed for identification, assay, water content, related substances, friability in case of tablets, disintegration, dissolution in case of capsules and tablets, stability analysis for all the products, viscosity in case of gels and ointments or ophthalmics, suspensions, osmolarity in case of injectables, microbiology in case of injectables and ophthalmics, particle size analysis in case of ophthalmic suspensions, other testing as appropriate for the specific type of formulation. So in pharmaceuticals, the API, excipients and finished dosage forms are being tested for various parameters at various stages of the development and manufacturing. In this slide, we are going to see specifically what are the analyses being carried out during a API synthesis. So the identification and analysis of raw materials and precursors is an important step before a MPI synthesis. So for these raw material and precursors or the intermediates, we need to do chemical analysis for identification and other testing. Then we have to do a infrared spectrophotometry analysis for identification of functional groups of the raw material. Then the assay of the or content of the raw material through various instruments. Then the water content through Fisher titration. Then residual solvent analysis by GS, gas chromatography. Then finally the limit test for the various toxic elements like lead, ion, arsenic will be generally carried out for the raw materials we source for the API synthesis or the intermediate source for the API synthesis. There are some specific tests are used to evaluate the drug formulation at various stages of the development. So that includes the API and the excipients which is procured for the development process, that is called raw material analysis. So which will be tested for its appropriateness and quality before the development starts for the finished product. Then there will be during the development of the finished product, you can do the in-process analysis. For example, I am going to do a tablet dosage form. So once I prepare the granules of the tablet, that granules of specific weight can be tested for the assay whether it is containing the label claim of the product or not. Then the granules converted into tablet, then the uncoated tablet will be tested, then the coated tablet will be tested. So like that, in process you can do a lot of analysis, and during the granulation you can do the bulk density analysis. Like that, so many in process analysis will be carried out during the formulation development. Then some capsules will be tested for the before and after filling. Then stability testing will be conducted for the finished dosage form during the normal storage condition and accelerated storage conditions. So stability testing is nothing but you are monitoring the quality of the product during the age of the product or during the time period when it is stored during the use of the particular product. So that is called stability testing, and all the tests which we have seen in the earlier slides will be carried out during the stability testing for its assessment of the quality and robustness. Then finally, the shelf life will be determined based on the stability testing of the particular product. Apart from the active pharmaceutical ingredients, excipients, the packaging materials also will be tested for its quality and robustness. And the packaging materials are classified into primary packaging material, secondary packaging material, tertiary packaging material. The primary packaging material is direct contact with the product, like bottles, foils, caps, etc. The secondary packaging materials are nothing but the which is used to pack the primary unit or the pack insert, like show box, package inserts, etc. Then the tertiary packaging materials, which is the secondary unit pack contained in the tertiary packaging material, and like corrugated box, etc. So these are also must comply with some of the standards which is maintained for the pharmaceutical products. So for that, these materials will be tested and used during the development as well as the manufacturing process. So the packaging materials can be analyzed or may be analyzed for some of the parameters, including the visual inspection based on the checklist and the sampling report of the packaging material, sampling scheme of the packaging material as per the acceptance quality level. Then the analysis of packaging material and approval of the packaging material will be verified by the quality assurance department of the specific R&D or the manufacturing industry. So the packaging material will be chosen based on the several parameters, which is tested parameters supplied by the vendors. If required, the R&D or the manufacturing unit can do some test to confirm the quality of the particular packaging materials. So aspirants, in this module we have seen specifically about the pharmaceutical analysis and the application of pharmaceutical analysis in various stages of pharmaceutical product development, including the active pharmaceutical product synthesis and development and manufacturing, as well as the finished product development and manufacturing. And also we have seen some of the specific testings required for the API, excipients, finished product, as well as the packaging materials. Thank you.